Well, hello there. It's Bruce Williams again, and today I want to start a series of lectures on ultrastructure. I've been giving this lecture for about 30 years, and for many years it was a mainstay of the AFIP and now the Davis Foundation's Descriptive Veterinary Pathology course. That was back when the ACVP valued description and ultrastructure. On my exam, all the way back in 1992, we had 2 of our 20 slides that we had to describe. Yes, 20 slides were ultrastructure, and we would describe them, we would analyze them and diagnose them just like any other slide. And I think it made us better pathologists. So this lecture is sort of focused on descriptive techniques, but I think there's a tremendous amount of good basic general ultrastructure information in it. It's not going to make you the world's best ultrastructural pathologist, but I'm hoping it will give you a little comfort when you come across photomicrographs of electron microscopic sections. A little more comfort, a little more confidence, and then you'll realize it really isn't that much different than what we see on the microscope. I like to say that it's like having a super duper objective on your scope. You know, we have all the regular objectives, including oil, which gives us a 1000x magnification. But imagine if you could spin it one more time and go to 10,000 or to 50,000. The same cells are there, the same organelles are there. We just get to see them up close. I think a lot of people are afraid of ultrastructure because it's in black and white and gray, and not in the familiar pink and purple. But as we go through the lectures, hopefully I will help you make that jump from pink and purple to black and white and back again, which will help you in your analysis and interpretation. Morphologic descriptions of an ultrastructural photograph are the exact same as a regular description that you would do for a slide, for those who are familiar with that particular process. There are a couple of differences. I will couch some of these slides in terms of that. The first thing that you want to do with a description of EM, or even any analysis of EM, is right up front we state the tissue or the cell that we're looking at, because usually you're looking at 1 or 2 or 3 cells, not an entire tissue. So it's important to let your audience know, based on what you see in the photomicrograph, what organ you're in, what part of what organ you're in, and what particular structure you may be looking at. I've always used a 3-paragraph system versus the 1-paragraph system for glass. I've used a 3-paragraph system for describing ultrastructure. And in the first paragraph, and you can cram them all together if you want, I suppose. I just like the little white space in there. The first paragraph is always the normal findings. This is what is probably the most different from describing EM versus describing glass. Once again, you're only describing a couple of cells. It's a little bit more of a specialized interpretation. And so, especially for grading purposes, if you are trying to convince the person who's reading this you know what you're talking about, a good thing would be to describe how many of what cells you're looking at. What are some basic features? Is the cell cuboidal? Is it columnar? Is it polarized with microvilli on one side, like you might see in the intestine? Are the nuclei located basally? Are there lots of smooth endoplasmic reticulum or rough endoplasmic reticulum like you might see in a cell that is producing a lot of protein? I tend to list all the organelles I can identify in this first paragraph to give as much proof as I can that, yes, I'm looking at a pneumocyte, or I'm looking at proximal convolute tubular epithelium. Okay, my second paragraph, because we're often asked to look at cells in various states of degeneration. Nobody really wants to show us EMs of normal tissue, so it's always some state of degeneration. So I will describe the abnormal findings that point me in that direction. whether it is lysis of chromatin, loss of microvilli, dilation of the smooth endoplasmic reticulum, all things we're going to get into in the second lecture. And then finally, my third paragraph are going to be the things that normally shouldn't be there. This might be infectious agents, it might be viral particles in the nucleus or bacteria in the cytoplasm, it might be abnormal compounds like internuclear inclusions in lead toxicity per se, which have a very characteristic look on EM. So it's things that aren't supposed to be there. You can't describe something that's supposed to be there and that is not. And there's something, because we're so up close, we're going to talk about later on in this lecture about when do we say something's missing and when do we say it's not. I've found that over 30 years, this is how I've approached it. It's always worked for me. I'm not going to say that's the best way or the only way, but it's worked pretty well for me. One of the things that sort of puts people off is they believe that there is a special vocabulary to ultrastructure, and there really isn't. Most of the terms that you're going to use, you already know, and hopefully as we go through these lectures, you'll realize that. There's no real special term. You may learn a couple of new terms along the way for structures that you only see when you get super duper close. But the vocabulary that you start this lecture with is largely going to be the same that you finish the lecture with. As I've graded many papers over the years, both for people describing glass and people describing EM, I've recognized that there are trouble words, which are sort of red flags that go up when someone's really not sure what they're talking about. Like when they talk about bronchi in birds or reptiles who don't have those structures, and it says, bing, this person's in trouble now. And unfortunately, you look a little closer at the rest of it, because if you don't know the normal anatomy, you probably don't know the pathology either. There are trouble words that I see in EM, and this is more for people who are grading EM. But when I see terms like structures, I know somebody really doesn't know the terminology, and so it's a red flag, and I'm going to look a little closer at that paper. Vacuoles, I do occasionally use that, but rarely. Most people, they see something that looks like a little clear hole and they say it's a vacuole. That's a term we learned back in 9th grade biology, and let's leave it there, okay? You should be using terms like smooth endoplasmic reticulum, which is clear. How about a lysosome or a phagolysosome? Occasionally, you can use the term vacuole if a particular agent, like a virus, say RNA virus, like coronavirus, has modified either smooth endoplasmic reticulum or lysosomes to become replication factories. And at that point, vacuole might be appropriate if you can't tell which is which. But generally, when I see that, another term that I see all the time with people that are just very uncomfortable with EM is when they describe there is a lot of electron dense material, or there is electron lucent material. You're not describing anything. You're not describing organelles. You're not describing pathology per se. You're basically commenting on the preparation of the material. Okay, if the technician's fairly new to this and they have too much osmium in it and it sticks to everything, you're going to get abundant electron dense material. A lot of stuff that is really heavily electron dense may be extremely degenerate if it's in a cell, maybe necrotic material if it's outside of a cell. Necrotic material really picks up osmium, and it just looks black. This is why you will never describe necrosis on an EM, because it's just a big black blob. It's all covered with osmium. So if you ever find yourself describing necrosis on an EM, chances are you need to back off and get a better section, find another spot on the grid where you'll be able to get some information that will help you. When we come to the morphologic diagnosis for an ultrastructural micrograph, like we do with every glass slide, we always formulate a morphologic diagnosis for ultrastructure for EM as well. I tend to keep it very simple because you're looking at one or two cells. You can't talk about a necrotizing hepatitis from two cells. It makes no sense. So I keep it appropriate for those cells. As we said before, you can't describe necrosis, so almost every time the cellular degeneration will be part of that. So if I was looking at the liver of a chimpanzee with adenovirus, I may have 2 or 3 cells. I may have several cells, but because of the damage the adenovirus does, especially in chimpanzees, you often only have 1 or 2 cells that are left. So I would just simply say liver, hepatocytes, degeneration, moderate, marked, severe, whatever you want, with intranuclear virus particles. Keep it simple. Keep it short. When you start to try and interpret too much, you're probably going to get into trouble. If you're looking at a neoplasm, you're going to have 4 or 5 or 6 cells that all look alike. I just put the organ, liver, colon, lymphoma, or something like that. Very simple diagnoses. Before we launch into the lecture proper, let me give you 1 or 2 other bits of advice about ultrastructure. I'm someone that gets a fair amount of ultrastructure brought to me. Hey, what do you think? And what I see are errors generally in the same area over and over, or maybe slight variations in the same area. And this is people that want to overinterpret what they see. We're going to look at some slides which have organelles that may be misinterpreted, and it's a very common thing. The first thing to prevent overinterpretation is make sure that you're not just taking really high magnification images. Most people, when we get a good virus, they just want to zoom in on the virus and take some wonderful viral pictures for publication. But don't forget to start at a level where you can see not just the virus, but then you can see the damage it's doing to the cells. So you want to get the whole cell in several pictures. And then you want to back out again to take a picture of that region of the tissue and have enough cells so you can tell, and maybe some normal cells, so you can tell what tissue you're in. Okay? A lot of the pictures I get, people have zoomed so far into the cytoplasm that everything becomes a bunch of little dots, and then they want to say, well, this looks like this dot in this article, this dot in that article. So make sure you have a range of pictures far away, medium distance, and close, and not just zoom in. The other thing that people want to do is they want to try and overinterpret really bad tissue. Now, the best EM is taken from tissue that is fixed for EM, whether you're in Michael's media or glutaraldehyde. This is something that's harvested and put right into an EM fixative. Okay, one of the biggest problems with EM is you can certainly prepare EM from a paraffin block, and we do it, and everybody does it all the time. But there are artifactual changes that happen to a tissue that's paraffin embedded that become ultra magnified like everything else is on EM. And having looked at a lot of paraffin embedded tissue over the years, some of the artifact can make them almost uninterpretable. And when you have a tissue that has a tremendous amount of damage, let's go back to that chimp liver with adenovirus, and so many of the cells are destroyed, and then you take it out of a paraffin block where you, even on the best of the cells, you have large areas of artifact, or areas where the osmium doesn't cover on an even basis. So people want to interpret these. And I would say that 9 out of 10 images that come to me, people are making interpretation on either stain artifact or something that they are too close or is too damaged as the result of processing to properly identify. Misdiagnoses are a lot more common than non-diagnoses in EM. I generally try not to overinterpret. And people will say, well, why did I bring it to you? That's so obvious. Well, because I don't want to make the wrong diagnosis. I've made a lot of them on EM over the years, and I don't want to make a diagnosis that's based on something that I really shouldn't be looking at. I would like to say that all the EMs in the world are great. They're not. EM is a very difficult thing to do. Finding the beautiful images that you see in textbooks are literally hours worth of work, or maybe the most serendipitous thing that you pop down on the right field. But it's not common, and I've spent hours in the EM lab looking for that one good image. And to think you're going to go in and spend 10 minutes and get a fantastic image, you may be deluding yourself. So those are just a little bit of advice from 30 years of taking EMs, looking at EMs, and thankfully I've never had to process it myself. It's a very specialized field, not something that your average histotechnician can do very well. So with that, let's start looking at some ultrastructure. Okay, so our first lecture is going to be fairly general. We're going to look at just the prototypical cell, nothing special. I want to look at some of the structures that you need to be familiar with. And I think you're going to find that there are very few new ones here, maybe one or two, or maybe we're going to change the terminology a little bit as we get super duper up close. So here is a diagram of a prototypical epithelial cells. Now here's some structures. It's easier to start out with some diagrams, but you know, epithelial cells have a polarity. They sit on a basement membrane, which is often trilaminar, as you see here, and the other end of the cell is festooned with a variety of membranous structures, including microvilli. All epithelial cells have microvilli. Some may have cilia. Now there's a paper that came out a number of years ago that says every cell in the body, whether it's epithelial, whether it's round cell, whether it is a mesenchymal cell, has at least one cilium. Now I don't know from that. I'll take it at face value. Okay, they all have one cilium, but some have more, a lot more cilia, like we would see in the respiratory tract. So they would be up here too. Okay. Now, because so many of these are in apposition with the external environment. Remember, your gut, the lumen of your gut is the external environment. The lumen of your respiratory tract is really the external environment. You don't want things coming in between cells. Enterocytes are absorptive, so they will take in the nutrients you need through the cell membrane, through the microvilli. So to prevent things from getting down into the body in between the cells, all epithelial cells are tied together at the top by tight junctions, which we're going to look at just a little closer. It goes all the way around the cells. Underneath that, we have another reinforcement of cell-to-cell adhesion, which are belt desmosomes. Desmosomes are structures that have keratin, really tough keratin filaments. They tie and they bolt these epithelial cells together. And you can see, once again, it goes all the way around the cell. It's a pretty solid circumcellular structure. You will have, they're not the only desmosomes. You see desmosomes sort of thrown around here and thither and yon, and those are known as spot desmosomes. So you'll see those down below the belt desmosome, and we're going to look at pictures of all these in just a minute. Remember that all cells communicate. epithelial cells communicate with each other. They share various compounds, cytokines, or whatever. And so gap junctions are where these cells can talk to each other, can secrete signaling agents to each other, etc. They're not going to do it in the area of a desmosome. So you will see those a little lower. And then at the bottom, to anchor those cells, are hemidesmosomes, which you only see at the part of the cell which opposes the basal lamina. There are a number of diseases that are a problem, cause problems with hemidesmosomes, or when the immune system attacks the hemidesmosomes, such as the various forms of pemphigus, bullous pemphigoid, etc., which target those, result in the cell lifting off the basement membrane because it's lost its fasteners. Desmosomes are tough stuff. Keratin filaments are the toughest intermediate filament, and these are really tough. And as we look at cellular degeneration, those are usually one of the last things to go. Everything else in the cell is gone, and finally those will break and the cell will float away. So if we look at that on transmission EM, and I do want to go back. If a cell has a certain function, it's going to be oriented a certain way, and that's why epithelial cells are oriented. Mesenchymal cells, round cells, they don't have a particular absorptive function or secretory function or whatever, so they're happy to sort of tumble around and they don't care. But epithelial cells are usually oriented a particular way. So when we look at this on transmission EM, here we have microvilli, and we're going to talk a little bit more about the microvilli in a bit. They're always at the apical end of an epithelial cell. That epithelial cell is bolted in a certain way to be unidirectional, and these are always at the top, per se. This little density right here is your tight junction, and it's going to go, we tend to look at everything in 2 dimensions, but in 3 dimensions, it goes all the way around the cell right underneath the microvilli. So as we said before, nothing gets in between. Then we have the intermediate zone. This is where some of those gap junctions will be, and there's a little space here. So you can get not stuff from the outside, but maybe stuff that's secreted from the cell to move from one side or the other. So we move a little farther, we have this spot desmosome, and look how dark this is. Desmosomes are often some of the darkest things that you'll see, but they're always at the edge of the cell, and we're going to look at them a little closer in a minute, and you can start to see these little fibrils. And these are keratin fibrils that give it their strength, and they're bolted in here. Okay, and then some cells, to have a increased amount of contact with their neighbor, will have these interdigitating membranes. Not every cell will have it. It depends on what type of epithelial cell that you have. They're also called plications. I haven't used that term in years and years, but you might see that. And there are certain neoplasms, for example, which you would diagnose, like nerve sheath tumors, that have this incredible interdigitation between cells. So I don't use this for much. I might note it and just move on. If you'd like to look a little closer at that, okay, here's your microvilli. Remember, especially on a test, you're not going to see a little M there. That is something that was put in this particular publication. There's a tight junction. Nothing's going to get through here. A little bit of a gap junction here, or intermediate zone. And I said we'd look a little closer at the desmosomes, and you can see that they're most highly condensed right at the membrane. But if you look closely, you can see all of the keratin intermediate filaments projecting into this, projecting across the space between the cells even, to give that incredible density and strength. Now, you will hear, and for the vast majority of cells, you'll only see desmosomes in epithelial cells. Think about it. What is the immunohistochemical stain that we use on epithelial cells? Keratin, because they're full of keratin intermediate filaments. That's what makes desmosomes. but there are certain exceptions. And lower vertebrates like fish and reptiles can form desmosomes between macrophages, between histiocytes. They can have desmosomes on the macrophages. This is a granuloma. This is the wall of granuloma. And remember, we use granulomas when we want to wall something off and not let it get into the rest of the body. And so lower vertebrates have developed the ability to put down intermediate filaments in their macrophages to create an even more impenetrable barrier. This is a picture from a fathead minnow that has a mycobacterial granuloma. We look at the bacteria here, and they're not very interesting. Bacteria are sort of boring on EM, but we're going to spend a lot of time talking about them later on in the lecture series. And here are some very nice desmosomes. These aren't cut very well up here, but it helps the body wall off those bacteria. Okay, so now let's look at the structure that everybody knows, the nucleus. Okay, so here's a nucleus. We happen to be in a plasma cell because we're surrounded by lots and lots of rough endoplasmic reticulum, which we're going to talk about in a minute. For some reason, somebody stuck a 9 on all of the mitochondria. I don't know why. There's a 4 on nucleoli. So the nucleus can be in round cells, in mesenchymal cells. It can be anywhere in the cytoplasm. In epithelial cells, they often tend to be a little lower down in the cell, especially if the cell is secreting something, which tends to be up near the apical membrane where it's going to be secreted from. Remember, it's not going to go sideways. It's not going to go down the basement membrane. It's always going to be up top. So I think in epithelial cells, your nucleus tends to be a little bit toward the base. That is, if you're not holding the EM upside down. So every living cell has a nucleus. If you're not looking at that, then maybe you're looking at a part of a cell, like a platelet, or as we get into cellular degeneration, maybe you're looking at a fragment of a cell, but they all have a nucleus. Now, I've seen people describe, and this is a very important concept, I've seen people describe nucleus as a very large nucleus or a very small nucleus. Now remember, when we take sections for EM, they're incredibly thin. They're much thinner than they are for our typical 5-micron segment for a microscope. And so you can get many sections and views through the same cell. And if you think about it, if you have a cell with a nucleus in the middle, if you cut it at the edge of the cell, the nucleus being around is going to be much smaller if you cut it by the edge of the cell than straight through the center of the cell. So the size of a nucleus is often very dependent upon the sectioning, how tangential you are. And so calling a nucleus small versus large really has no moment unless you know, which you never do, that you're getting a slice right through the center of the cell. Nuclei have two types of chromatin. They have this dark heterochromatin at the edge. This is junk, I call it junk chromatin. It's stuff that the cell's not using. It might be for stuff that this cell's never going to make. It might be damaged chromatin, but it's junk. And so it accumulates at the edge of the cell membrane. You don't translate any RNA there. It's just useless. And then you have euchromatin, which is where all of the RNA is being translated and proteins being made, and all of that is unfurled chromatin to make protein templates. That's the useful chromatin. Euchromatin, useful chromatin. Now, a lot of people like to spend a lot of time saying the nucleus is small. The nucleus is small and has large amounts of euchromatin and small amounts of heterochromatin. I'm like, oh, you told me nothing. Okay, everybody knows euchromatin, heterochromatin. I never can assign points for that if I'm grading out a description. People like to write it. It really doesn't make all that much difference. I can tell very little from a cell from the nucleus itself. I can make a very general interpretation here if everything's preserved well. Cells that have a lot of euchromatin, you can do the same thing on glass. Cells that have a lot of euchromatin and prominent nuclei are probably making a lot of protein. They're active, metabolically active cells. Cells that have a tremendous amount of heterochromatin, like plasma cells or whatever, they make one type of protein over and over, or they're not very active at all. And that's as far as I'm going to go with it. I'm not going to look at that and try and make any more interpretations than that. I want to keep it simple. If you look a little closer at the edge of the nucleus, you will see the nucleolemma, or the nuclear membrane, or the nuclear envelope. Okay, this is a connection to the smooth endoplasmic reticulum. A lot of the cytoplasmic mediators that are coming out of the nucleus enter the cytoplasmic reticulum this way. Now, it's not totally complete. There are holes in it which are known as nuclear pores, and if you cut that nuclear membrane on face, which means you cut right through it, okay, what you're going to see are these tiny little nuclear pores. So that is how proteins and RNA and other agents of communication with the rest of the cells get out of the nucleus into the rest of the cytoplasm. Okay, so they come out through nuclear pores. And then there are parts of the nuclear envelope which are stuck to the nucleus. So it just sort of disappears in spots. Something that is cool about the nuclear membrane, because it connects to the smooth endoplasmic reticulum. We're going to look at this later. We'll talk about degeneration. Is that if you have damage which causes influx of water into the cell and dilates the smooth endoplasmic reticulum, you may look at that and you may say, is that really dilated, or am I overinterpreting it? But if you see expansion of this nuclear envelope, which is connected to the smooth endoplasmic reticulum, I've never seen that in a cell that wasn't degenerating. That's one that I can hang my hat on and say, yeah, that cell's having some problems. It's taking on water. So I use it for that. Okay, nucleoli. Most cell nuclei have nucleoli. There are two parts to the nucleoli if you want to get deep into it. You have the granular or lucent areas of nucleoli, and you have the fibrillar part. I just don't use that very much. Okay, I just say that the nuclei are here or they're not. Now, when you have degeneration of the nucleus, generally the first to go are the nuclei. They just sort of disappear. I don't think that you can say there's no nuclei, this cell is degenerate. You can say that there's no nucleoli. And if you have other features of cellular degeneration that go along with it, maybe you can make an interpretation. But I simply note the presence or the absence, and that's about it. So they're cool, but they're not very useful in terms of ultrastructural description. Now, probably the most useful, or at least one of the most useful organelles when it comes to talking about ultrastructural anatomy and pathology, is the mitochondrion. And the mitochondrion is the perfect example of something I'm going to say over and over in these lectures, and that is form follows function. Form follows function. And you can look at a mitochondrion and you can actually make some decisions about the cell and the metabolic activity and what type of cell even you are looking at by looking at the way the mitochondria are structured. Now, the mitochondria on the outside, like all organelles, has a double lipid or a lipid bilayer membrane. Okay, now you think, well, lipid bilayer, that's really a cell membrane type thing. Well, it's all the organelles too. And compounds that damage the lipid moiety of the cell membrane often do similar damage within cells, and that's where we get lamellar bodies. And we're going to talk more about that later on when we get to cellular degeneration. So we have a lipid membrane on the outside, and that is continuous with the cristae here. And the cristae, which are nice and long, and there's lots of them in this particular cell. So that tells me, because cristae are where oxidative phosphorylation takes place, where ATP is made. The more cristae you have, that means a higher need for ATP. Those cells are more metabolically active. They, by nature, have a higher oxygen requirement. We see particularly long and lush cristae in cells like muscle cells, or the proximal convoluted tubule, something where it takes a lot of oxygen, a lot of ATP is expended. You're thinking, well, I know why a muscle needs a lot of ATP. Why does renal tubular epithelium need it? Well, remember, they have a pretty profound secretory function where they're often secreting compounds against the gradient, and that takes energy. You'll see long cristae in the mitochondria of the stomach, for example, where you're making acid and secreting that against the concentration gradient. Okay, we're going to look at some cells which have very short cristae. I call them couch potato mitochondria. They're just not doing much. In between the cristae is the matrix of the mitochondria, and this is where anaerobic glycolysis takes place. So cells that use a lot of anaerobic glycolysis, they don't have a high need for oxygen, like cells, believe it or not, within the central lobular liver. They don't normally get a lot of oxygen, so they tend to have sort of runty, stunted cristae. Okay, they don't see oxygen, they've evolved not to use it and to use a lot more anaerobic glycolysis. Okay, remember that mitochondria has mitochondrial DNA as well. You can't see that, but the site of transcription is also in this granular matrix. Most mitochondria have some calcium deposits in them, little black ones, and if you have a couple, that doesn't mean anything. We'll see that as they degenerate, you get a lot more of these calcium densities, and it can be, with large numbers of them, a sign of mitochondrial degeneration. So remember, the more cristae, the more robust the cell. And this is one of a number of little bits of evidence we have to put together later on. We're trying to figure out what type of organ that we are describing. Now, cristae, this is sort of your normal garden variety cristae. There are also some mitochondria which have these sort of bulbous swellings of the cristae when they're cut in. They're not really swellings, they're just bulbous. These are known as tubulovesicular cristae when they're cut a particular way. And you see these generally in steroid producing cells. Like you might see them in the adrenal cortex, you may see them in the interstitial cells of the testis, the Leydig cells. And you can also see there are a lot of smooth endoplasmic reticulum. But this can be a clue that you're looking at a steroid-producing cells when you see these particular cristae. So I always, when I look for mitochondria, they're easily identified in most cells. I look for the number of mitochondria. Cells are metabolically active, going to have a lot of mitochondria. They're going to have a lot of cristae, so they can produce a lot of ATP. Okay. Rough endoplasmic reticulum. Or we'll just start with endoplasmic reticulum. I think you already know, many of you already know that there are two forms. There are the rough endoplasmic reticulum, which are studded by ribosomes. And this is where a lot of protein is made that will be exported from the cell. If you look in between some of this rough endoplasmic reticulum, you will see ribosomes, which are known as free ribosomes, which are just floating around in the cytosol. And that is the site where protein is made that stays within the cell, a replacement for cell membranes or parts of organelles or whatever. So everything that's made in the lumen of the rough endoplasmic reticulum will eventually be exported from the cell in the form of a cytokine or antibodies or something like that. So cells that export protein, like plasma cells, have a lot of rough endoplasmic reticulum. We'll look at that as we go farther down. Cells with very little, they don't make, like neurons, they don't excrete a whole lot of stuff. Every cell excretes something. But some, like neurons, they're not made to excrete a lot except for maybe some neurotransmitters. So you don't see a lot of rough endoplasmic reticulum. The other thing is we start to, and I said we talk about pink and purple and black and white. I want you to start making this leap in your mind. When you have tremendous amount of ribosomes, it's a tremendous amount of protein, and most H&E stains will stain that purple. So I want you to think about this plasma cell right here, and it's just full of rough endoplasmic reticulum. We looked at one earlier, and you could see it's just layers upon layers upon layers of rough endoplasmic reticulum. When you think about plasma cells, what color are they? They're purple. So anywhere that you have a concentration of ribosomes or rough endoplasmic reticulum, that part of the cell is going to be purple. It's going to be the protein producing part, and it's going to be a deep purple. Here's a poorly preserved mitochondria with low calcium density. I can't tell much about what's going on here. I don't really see the cristae as well, but this, we're just talking about the ribosomes. So we'll look at it in a minute. Sometimes you will see aggregates of glycogen within cells which are about the same size as ribosomes and can be confused. But usually you only have glycogen in certain cell types. I skipped one. Okay, now let's start to put a couple of these things together. Here is a section from one cell. We have a very tangential piece of a nucleus. I can't tell how big this nucleus is from that. I'm not even going to try. But the nucleus is, as I describe this, I'll describe all the things that I can identify. I can identify a part of the nucleus. I can identify a couple of mitochondria, and let's look at these mitochondria. Okay? Very few cristae. These are classic, what I would call couch potato. They're not making a lot of ATP. So it's a cell that usually doesn't get a lot of oxygen or can do its job without getting a lot of oxygen. Okay, here is, and it's easy because we have SER here, but this is smooth endoplasmic reticulum. Not a lot of ribosomes. I can see some clusters of ribosomes perhaps, or glycogen here, but there's a lot of smooth endoplasmic reticulum. This is actually too much smooth endoplasmic reticulum, but it's a nice picture. This is a liver cell. And remember, we talked about liver cells, especially in the center of the lobule. They are literally the last cell in the body to get oxygen. The blood goes all the way around the body, and then right before it goes back to be reoxygenated, it visits the central lobular hepatocytes. So they never get oxygen. Okay. The other thing that they will do is that is where a lot of toxins are biotransformed, or drugs are transformed into innocuous agents. We hope they're innocuous. And if this happens over time and you're getting a lot of drugs, you get a lot of hyperplasia. This particular picture was from a dog that had been on phenobarbital or phenytoin, another anticonvulsant, for a long time. And this cell had actually evolved to have more SER so it could detoxify more and more. But it all is starting to make sense, and that's what I want you to have, these little light bulbs that go off. We have a nucleus, we have couch potato mitochondria, which you would see in cells that are usually hypoxic, and lots of SER. So this is, I think there's enough information you could say that this is from the liver. Okay, there is some rough endoplasmic reticulum. Every cell has to make some proteins that are kept behind. Okay, but this cell is primarily for detoxifying. And then there are these things. See these little black things? That's garbage. That's trash, and don't interpret it. If it's really black, you can't tell what that is. Here's something that looks, there's some black, there's some white. Could it be a lysosome or phagolysosome? It could. I tend to try to stay away from trying to interpret this stuff. And I don't think that saying there are multiple electron dense structures that red flag word is helping us in our description. So I tend to leave this out. I stick with the stuff I can identify. If you're lucky enough to have letters here, here is a G, and that stands for Golgi apparatus. And the Golgi is a very important organ. It is like the post office of the cells. And protein that leaves the nucleus generally goes into the Golgi, where it is addressed. They put an addressing on, and that tells it where in the cell or outside the cell it's going to go. Maybe it'll put one on, says you're going to go up to the cell membrane and make a patch there, or you're going to go into one of the mitochondria. And all that happens in the Golgi. The proteins are what's known as transitional vesicles, and they move from the cis face of the Golgi to the trans face. They can actually move backwards in retrograde vesicles. But the concave, and I think I had that wrong. I think there's a cis face here, which is the concave, and then the trans face here. So it puts glycosylated moieties on the cells and addresses proteins for where they go. This convex face is always facing the nucleus. Not that it really makes any difference, because if we were a little farther away, you would be able to tell that. In certain cells, they're very prominent. They're usually not terribly prominent. Certain cells, like plasma cells, they form what's known as the HOF. It's that cleared area in that sea of purple endoplasmic reticulum. And they're big, and they're busy, telling all the proteins, you're an antibody, you're going out of cells. You're an antibody, you're going out of cells. You're an antibody, you're going out of cells. But they probably have the most prominent and important Golgi of any cell in the body. Okay, so to this point, I don't think I've given you a single new word for your vocabulary. And for a lot of you, I'm not going to give you any on this. This might be the first time that we put anything new in our vocabulary. And I've told you before, certain rules. Try not to interpret anything that's really black. And also don't use words like vacuoles. That just it grates on my nerves. I will use it once or twice, very specialized circumstance, which we have not come to. Now, we've looked at mitochondria here. I'm looking at this one. It's sort of half and half. It's got some nice long cristae, not a lot of them, so I can't tell much about that. I've got rough endoplasmic reticulum here. I've got a lot of smooth endoplasmic reticulum. Usually, in just about any EM, you'll be able to pick out smooth endoplasmic reticulum. It's always a little swollen in certain areas because just the processing. Okay, just the processing. But now what I want you to look at is I want you to look at these structures here. They're round. Some are fairly homogeneous. If you look really, really closely, and I don't know if you can get close enough on your computer, they also have a little double membrane. Every organelle does. Okay? And this one is homogeneous, and then some of them have these blackish blobs. And as we get farther into this, one of the things I'm going to say is that a compound in the body that really takes up a lot of osmium always stains very black. If it's really there, is fat. Fat's a very interesting thing on EM. The processing of tissue for paraffin embedding removes all the fat. Okay? So if you're taking tissue out of paraffin, the fat in that tissue, if you're looking at a liposarcoma or something, it may stain just totally clear because all the fat's been removed. It may stain incredibly black. If you did it properly and you're using a real EM fixative, it's going to be very black. Osmium just sticks to fat or anything that contains fat or fat breakdown products. Okay, so these little homogeneous ones are lysosomes. These ones with fat in it, and I'll flip all the cards on this one. This is lipofuscin. Remember, it's a fat breakdown product. These are what we're going to call secondary lysosomes. Okay? So these are primary lysosomes. They form with structures which look like this. This is a multivesiculate body, and multivesiculate bodies, remember, the cell membrane, organelle membranes are always wearing out, or they have certain receptors that the cell no longer wants or needs, and they're resorbed, or you have little penocytotic vesicles, which are parts of the membrane that invaginate. And so the body is continually, or the cell is continually reinvigorating itself, putting out new membrane and taking out effete parts. And they end up in these phagosomes, which when you have these little bits of membrane, remember membrane is almost all fat, they're called multivesiculate bodies. And what will happen is this multivesiculate body is going to fuse with one of these primary lysosomes, become a secondary lysosome, and then the proteases and the lipases in the lysosome are going to break down that fat, give us something that attracts osmium that will look like a brown granule underneath the glass or under the microscope, and will look like black blobs. And this is a secondary lysosome full of lipofuscin. Okay, so you will see this in cells where there is a widespread injury to cell membranes. So you may see this in cases of vitamin E deficiency. You will see this certainly in conditions such as ceroid lipofuscinosis or intestinal ceroidosis. A lot of the things which used to be called lipofuscinosis now are ceroidosis because it's a pathogenic process rather than a natural breakdown process. So anything that injures cell membranes, you're going to see a lot of this material. Okay, let's do a little detective work. Mitochondria, very few cristae. Okay, a lot of matrix. Couch potato, mitochondria. I'm already thinking of liver. Anything else that's going to help us with liver? Well, here's a big round thing of fat. You see a lot of fat in liver. Then the other aggregates here, this is what I want to bring up. These aggregates are known as alpha particles of glycogen. Glycogen comes mostly in alpha particles. It's sort of bundled up. There's also beta particles which are individualized, which could just as easily be ribosomes to my eye. Maybe here's one here, here's some here. We're up near some rough endoplasmic reticulum. So I think without the alpha particles or the glycogen aggregates, it's really tough. But when you start thinking, okay, cells that contain glycogen, fat, and couch potato mitochondria, this has got to be liver. So this is the way we're going to progress through these lectures is we're going to take what we're given, we're going to think about it logically, and we're going to come to the magical diagnosis without seeing anything more than an eighth or a tenth of this cell. I'd like to say it works all the time. It really is helpful with liver. We get in some of the more complex organs is not as good. But that's what glycogen looks like. And remember, only a certain number of cells even store glycogen, muscle and liver. And I think that's about it. There's probably one or two others. Let's look a little closer at fat. And this is the problem that you get when you use paraffin-embedded tissue. Everything gets torn up. Okay, you get a lot of holes, and then based on how long it was in the various graded alcohols and the processing time, your black could be anything from, or your fat could be anything from a big black blob to just a clear hole. One of the things that you will know about fat, regardless of how well it's stained, is that it does not have a lipid bilayer. It just floats free and it aggregates into these round droplets because everything in nature tends to have surface tension, and round is the default shape in nature. We're going to use that later on too, that little bit of information. But it pushes everything else out of the way. This is a bunch of glycogen, which has just gotten pushed out of the way. It tends to adhere to not really adhere, it just tends to be sitting on the outside. So fat tends to push organelles, glycogen, everything out of the way, whether it is a clear looking hole it is sort of grayish and variegated as this, or it is a dense black blob in this properly fixed specimen. Okay, so let's take a look. Now we've looked at most of the basic organelles, and we will probably throw in a couple of sort of rare ones farther down the line in some of the other lectures. Let's look at some of the non-organelle components of the cell that are important. And here is a freeze fracture preparation, which demonstrates, we're looking at the microvilli at the top of our prototypical epithelial cell, and you can see all of these little filaments that go up into the microvilli, and these are actin filaments. Actin is the smallest fiber in the cell, and it gives the cell essentially its body, its shape. It varies between filamentous forms of actin and the globular forms of actin, and the body of the microvilli are filled with these vertically oriented actin filaments, which go down through the microvilli, and you can see how much of that is in the cytoplasm. The organelles sort of are within and squeezed between and all of that, and then it terminates into this condensation of myosin, which is underneath the microvilli. It's a little thicker, with this being 6 angstroms, is about 10 angstroms in diameter, and this is known as the terminal web. You can actually see these filaments and the terminal web on very, very high transmission EM. It's not something that you really talk about much. It's not something that you would describe because it's one of the smallest things. You almost never get close enough, and really there aren't a lot of pathologic conditions which are associated with problems with actin polymerization. In cells, especially cells that are motile, it's about 15% of the total protein of the cell, which tells you how much is actually in the cell. So actin filaments and myosin. Microvilli, so we know that we're dealing with an epithelial cell. And just one more picture of the microvilli cut and cross section. You can see the actin filaments. Here is the tight junction, gap junction, desmosome, as we've already seen. Now it's very different. Microvilli look very different than do cilia. Every epithelial cell has microvilli. They're generally shorter, a limited number of cells, and obviously we see microvilli in the respiratory tract, where they perform an extremely important function in the mucociliary escalator. We can see them in a number of other organs, including the choroid plexus, including the gonads, including the inner ear. But cilia, instead of being primarily composed of actin, are primarily composed of tubulin. Tubulin are sort of interlocking blocks which can polymerize and depolymerize, and the shifts in polymerization allow the cilia to move back and forth. And cilia is something that is very well conserved in nature. The 9+2 arrangement of the Dynein arms is similar whether you're looking at a cilium from a protozoan like a flagellate or a ciliate, or whether you're looking at the respiratory cilia of a human being. It's the same pattern over and over, 9 outer doublets, 2 inner singlets, and we're just looking here at multiple cross sections through the same cilium. There are problems which are diagnosable via EM. This is a very classic picture that was actually on the ACVP board exam. So if you weren't familiar with this particular condition known as ciliary dysgenesis, you might be sort of messed up. But if you count, most of these cilia have the 9 outer and 2 inner, but then you start to realize that this one doesn't have any of the inner singlets. This one only has 1 instead of 2. Okay, this has 8. Well, I'm sorry, that's normal. This has 8 here. This one has two pairs of singlets on the inside. And the problem with this is that these abnormal cilia do not have the proper motion. They can't function like a normal cilium, and so these animals often have severe respiratory infections. They don't have a functioning mucociliary escalator, and it's often associated with another birth defect. And if you've seen any of my lectures, you realize when you get one birth defect, one congenital defect, you usually get multiple ones. And this one is often associated with a condition known as situs inversus. Situs inversus is where all the organs of the body are flipped. And so normally the stomach would be on the left-hand side, but it's on the right-hand side. And it's a mirror image. So those are, I'm sure that those didn't get diagnosed for a long time, because you don't normally look inside an animal and say, everything's backwards. And then obviously this is one that you can only diagnose from EM. But the connection has been made both in humans and in animals, and it's a well-known condition seen in certain inbred strains of mice, and then occasionally in other animal species. Now, there is a structure at the bottom of every cilium, which is known as a basal body. There is a condensation at the bottom, diagrammatically. Well, here's one right here, and it's known as a basal body, and essentially anchors the cilium and has a function in motility. And a very similar structure appears only close to mitosis in the cells. This is the mitotic spindle, or the base of the mitotic spindle, or the centriole. It actually, to me, looks like a little turtle, or maybe a beaver, if this is the head and this is the tail, and it always appears right before mitosis in the area of the nucleus. There are actually two, let's use the term basal body for lack of a better word, in the centriole, and these are anchors for the mitotic spindles. They're oriented at a 90-degree angle to each other, and so the spindles connect to the separating chromosomes here, and here they contract and they pull apart. So it's very rare that you would probably ever find a centriole, but it has the same arrangement of singlets and doublets, the 9+2 arrangement that we see in cilia. So cilia, basal bodies, centrioles, okay, all interrelated organelles, and the centriole is one that you only see at the around mitosis. Another filament that, oh, here it is right here. What a great picture. And here is your centriole. It has all of these microtubules which are attached to the dividing chromosomes, and they will retract. Okay, so talk for just a minute about the microtubules. Microtubules are considerably larger, being around 25 angstroms as opposed to 6 angstroms for actin. They're not as many, certainly. And when we talk about the motility of organelles, organelles are not just floating free in the cytoplasm. Okay, they are anchored in certain positions. Mitochondria may be in highest concentration in the areas of the cells that or have the highest requirement for energy, such as the near the basement membrane in tubular epithelial cells. So something has to get them there. They're just not floating free. We have this picture in our head of cells being just a big bag. And it's really not like that. There is function and structure and directed motion of organelles. How do little pockets of proteins get out to the cell membrane? How do pinocytotic vesicles get in from the cell membrane? So there is directed motion within the cell, and it's accomplished via the use of these microtubules. I call them the railway of the cell, and they're difficult to see. You're very lucky if you do see them, but here's a great picture of microtubules, and they go all over the cells. And I like to use the analogy of this mitochondria is traveling along this, but they are required for any type of secretion of granules. Any shape change of the cell requires microtubule formation. There are certain toxins which poison microtubules and paralyze cells, like colchicine, and they do that by causing a disassembly of the microtubule filament. So it, along with actin, are probably the most important filaments of the cell. Actin giving the cell body and form, and microtubules providing the ability to do many, if not most, of cellular functions. So those are all the things I want to talk about inside the cell. We have just a minute. I want to talk about, I think, some of the extracellular structures that are commonly seen in electron micrographs. You should be able to identify. They're often associated with normal or pathologic processes. The first one that you will see in the extracellular space is collagen. Okay, notice a couple things about collagen. Here's collagen fibers which are cut in cross-section. Here are collagen fibers which are cut in longitudinal sections, and you will see, if you look very closely, there's a very distinct and regular periodicity. There's this periodicity too, so this long black and white, but that's not the one I'm talking about. I'm talking about these little black stripes, and they're precisely about 64 angstroms in length, and it's a very regular thing, and that's one of the things that helps you identify collagen if you need to. I don't think it's difficult to identify. It's often in the extracellular space, especially when you're dealing with tissues, not epithelium, but maybe a little bit deeper. Very commonly seen. Sometimes, as you'll see, we may diagnose certain spindle cells as fibroblasts because they are surrounded by so much collagen. It's very interesting. Collagen is polymerized in the extracellular space. You have 3 strands of which go together to make one mature strand of collagen. You can't see these in the cytoplasm of fibroblasts. They look like any normal cell. It's only when they're in the extracellular space that that it polymerizes and forms true collagen. But I think collagen helps you to establish that you are, in deep in a tissue, maybe in mesenchymal tissue or something like that. So it's certainly worth looking at. There are a number of conditions that result in abnormal forms of collagen. Ehlers-Danlos syndrome or dermatosparaxis, which results in variable sizes of collagen and fragmented collagen. There are a number of specific conditions where you have short-spaced collagen or long-spaced collagen. Some people call these zebra bodies, and they tend to clump up. Here's your normal collagen here. Then these are the abnormal ones. They're seen in a number of pathogenic conditions. Now here's something that is fibrillar. It's in the extracellular space, okay, and it just forms these sort of dark, densely packed globs. When you look at collagen, it's sort of arrayed in the same direction to give tissue tensile strength. When we look at this has a periodicity as well. We don't have a measure, but it's about half that of collagen, about 23 angstroms, and it's very dense. The fibrils are, for the most part, just put down willy-nilly, like haystacks. And this is fibrin. And you can imagine why you would want all of your fibrin strands to go in one way, because stuff would leak in between. So it's put down on top of each other. So this is what fibrin looks like. And we'll have a couple of slides later on of various conditions where you have leakage of fibrin. And you can see it tends to be very dark. And it's not that difficult to recognize that, especially if you have erythrocytes in the neighborhood and maybe some white blood cells. This might be a little difficult because we have nothing else to say, this is fibrin. So, but that's what fibrin will look like. And then the last one that I want to show you is amyloid. And amyloid, unlike fibrin, it is haphazardly arranged, but it doesn't have that dense packing. Amyloid fibrils are generally very loosely arranged. Here are some collagen fibers so you can get a nice size comparison. With this being the width of one of the collagen fibers, we see the periodicity. Amyloid does not have a periodicity. And if you're looking for the beta pleated sheet effect, you're not going to see it on EM. Okay? That's with some other form of viewing, spectroscopy or something else that I've never used in my life. But on EM, and we're probably in a glomerulus here, it could be in a liver or something, but you can see this material, expands and pushes everything out of the way, but it's very loosely arranged. So I think those are three fibrils that you probably should be able to recognize. Like everything else in ultrastructure, it's based on a lot of considerations, including hopefully knowing where you are. And if you're in the glomerulus and this was a dog who was proteinuric, sure, you see something like this and say, yeah, okay, we've got amyloid. Okay, well, that covers all of the basic organelles and some of the intra and extracellular fibrils and components I think that you should know for us to move forward. I hope I didn't teach you too much new stuff, because really, as we said before, the vocabulary and what you know from looking at the microscope is going to serve you very well for the rest of these lectures. We're about 75 minutes. I think we have a good background. We're going to move forward to the next one. Should take about a half hour for cellular degenerative changes, and I look forward to seeing you next time. Have a great day, and I hope you are happy and safe and healthy. And we'll see you again.